everybody. Welcome to Chapter 9. In Chapter 9, we're going to be talking about muscles and muscle tissue. And so let's go ahead and get started. So when we think about muscle tissue, we're talking about half of the body's mass, or nearly half. It does vary a little bit depending on how much you work those muscles and build those muscles, but it comes out to about half of the body mass. Muscle tissue is extremely important because it can transform that chemical energy, we've talked about it before, but ATP, adenosine triphosphate, into a directed sort of mechanical energy, which is capable of exerting force. Actually, just kind of a side note, I've had students in the past ask me, why do I need to understand physics, or why do I need to understand chemistry? Well, this is one of the things that why understanding chemistry and a little bit of physics is important, because you have to understand the chemical energy aspect of it, which is that chemistry side, as it's directed into this sort of mechanical energy, and that's sort of physics. Think levers and fulcrums and all of that. So just a fun side note to let's see the connection between all the sciences. But to investigate the muscle, we do have to look at the different types of muscle tissue there are and the characteristics of those different types of muscle tissue and how that muscle functions. And so that's what we're going to be doing in this chapter and even into the next chapter as well. So some of the terminologies that you see, myo or mes, and even sarco, is just sort of prefixes for muscle. So you're going to see that a lot. An example is sarcoplasm. That's a muscle cell cytoplasm. So that little gooey substance inside cells, when we're referring to that inside of a muscle cell, we call it the sarcoplasm. There are three types of muscle tissue. And we're going to go through each of these, but skeletal, cardiac, and smooth. Now, the important thing to know about these is only skeletal and smooth muscle cells are elongated and referred to as muscle cells, or muscle fibers, sorry. And then we're going to have cardiac muscle, which is going to be slightly different as we kind of talk about those, kind of going through. So let's compare them just a little bit. Skeletal muscle tissue, of course, is attached to bones. Some facial muscles are attached to skin, but in general, they're attached to bone. And they're very sort of this long, cylindrical, multinucleate cell with striations. So you see these striations, you see lots of little nuclei kind of aligning those. So that's sort of that what skeletal muscle will look like. Cardiac muscle, they line sort of they're part of the walls of the heart. And you can see it's got these little branching chains of cells. They're uni or even binucleate. They do have some striations as well, so you can kind of see some striations here. You can see some either one nucleus in a grouping or sometimes two in a grouping. And basically, you can see, compare that difference. Now, smooth muscles, it's usually what we call a unitary muscle that's in the walls of hollow visceral organs, other than the heart. And so you can think of stomach, intestines, things like that. This is going to line that. Also lines things like, it also is eye muscles, airways, large arteries, etc. Now, these are single spindle-shaped uninucleate with no striations. So you don't see these little distinct bands like you would see in that skeletal or cardiac muscle. And, of course, there's one nucleus associated with that. So that's some of those differences of these muscles we're going to be talking about. So muscles do have some characteristics, four main ones to be exact. Basically, one is excitability. That's the responsiveness, the ability to receive and respond to stimuli. If you need to pick something up, it needs to receive a stimulus and respond to the stimulus for you to pick that up, whatever object it is. Contractibility. They need to be able to shorten forcibly when stimulated, meaning I need to be able to pick something up. I need to be able to contract them or cause that contraction to happen. Extensibility, basically the ability to be stretched, and that should make sense. If you just take your arm now and bend and stretch and bend and stretch, you can kind of, get that extensibility aspect. Elasticity as well. It needs to be able to recoil to sort of this resting length. So if I stretch out my muscles and I'm just putting my arms out to the side, and then I kind of, let that go, that muscle has to have the ability to just go back to its resting state without any kind of damage to it. So that's elasticity. So that's really important as well. So muscle, of course, has functions. So there's four main important functions. One, it's going to produce movement. It's responsible for all locomotion and manipulation. So walking, digesting, pumping blood. So muscle function is essential. You can imagine any kind of genetic disorder or any kind of disease that messes or affects muscle functions can be devastating because it does all of these different things for us. The muscles do. It helps us maintain posture and position. That's also alongside as it's our skeletal system, but the muscles allow that to happen too. They help stabilize joints, which is super important in the overall process of, just getting from point A to point B and to, kind of maintain those body weights without damaging joints just one time of standing up, right? And, of course, they generate heat as they contract. That is a sensation, of course, you're probably familiar with. If you walk fast, go run up and down stairs or anything like that, you start feeling hot and in some case sweat. So that's the response of your body utilizing ATP as they're contracting back and forth and basically generating that heat that largely leaves your body. So skeletal muscle anatomy, kind of focusing specifically on the skeletal muscle. Skeletal muscle is an organ made up of different tissues with some features. Basically, one, nerve and blood supply. So there's nerve and blood supply to our muscles. There are connective tissue sheaths, and there are attachments to these. So we'll kind of work our way through these as well. Each muscle receives a nerve, artery, and veins, and they are consciously controlled. Skeletal muscles have nerves supplying every fiber to control activity. So if there's these muscles that we can think about and move, meaning we have control over them, basically they have that nerve supplying every fiber to allow that to happen. We will talk about some that, there are some muscles that we don't control, right? I mean, an example is we don't consciously control our heart, right? Yeah, it's working, it's beating, it's got nerves, it's got all kinds of stuff to it. But it just beats on our own. We can't go beat, heart, beat, and make it beat, right? So some we consciously control. And that's skeletal muscle. Contracting muscle fibers do require huge amounts of oxygen necessity and nutrients. And it also needs to have the ability to remove waste products and remove them rather quickly as well. And so that's another important aspect that muscles are going to need to do. So here is an image of that connective tissue sheaths that are present as well of that skeletal muscle, and sort of the layers that. So these connective tissue sheaths basically have three different layers associated with them. So we have the epithelium, the paramecium, and the endomecium. I've heard people pronounce it different ways, and it's perfectly right to do. I think some people use this mycium, and I say mecium. Either way is perfectly fine, and no one is right or wrong, and how you can say it either way. Anyways, so you can see when you zoom in, here on the outside you can see this epimecium here, but if you zoom in, you can see each of these different areas or these sheaths located in different places. Again, you also have blood vessels, so you will see those as well located there. Here, of course, here's where the tendon attaches that muscle to that bone as well. So attachments, of course, is important in this overall process. Muscles span joints and attach to bones. And so muscles do attach to bone in at least two places. We have an insertion, and that's the attachment to a movable bone. So you can think of, you definitely have an insertion if you can bend your arm back and forth. You know, that movable bone is going to be muscles attached. That's what we call an insertion. And then we have origin. That's attachment to a movable, to an immovable or even less movable bone, something that's difficult to kind of move. So attachments can be direct or indirect when we talk about how they're attached. Direct is more fleshy. So you have this, basically the epimysium is fused to the perosteum of the bone or the perichondrium of the cartilage. And then, of course, indirect. That's where you have this connective tissue wrappings that extend beyond the muscle, sort of like a rope-like tendon, or what we call sheep-like aponeurosis. And so those attachments, like I said, can be direct or indirect. So basically, here's that organizational level of that skeletal muscle that we've been talking about. So you can see, basically, here's that muscle, here's that epimysium here, there's a fascicle here, and so you can see muscle, and then there's that tendon. Those, of course, there can be hundreds or thousands of muscle cells, plus connective tissue all around that, blood vessels and nerves. You also have this facade, basically, that's a portion of the muscle, part of the fascicle here. And so you have paramecium, you have these muscle fibers, and of course the fascicle is just sort of this discrete bundle of muscle cells segregated from the rest of the muscle by either some kind of connective tissue sheath. It's really hard to say. Surrounded by that paramecium. And then, of course, you have that muscle fiber cell. And so you can kind of see, here's a nucleus. You do see some of these striations, at least in this particular image. And then you have these sarcolemmas, that endomyosin, a myofibril. So the muscle fiber is just an elongated multinucleus cell that is branched or striated in appearance here. And, of course, it's surrounded by that endomyosin. So when we talk about these muscle fibers, there's some microanatomy and some movement that we can think about, and we call this the sliding filament model. I will say there are some videos in the main PowerPoints attached. I suggest watching those. I will say the sliding filament model is very hard for some to understand. And it's hard for me to even talk about it in here because if I play a video in this, you won't hear it because of the way my computer is set up. So I just wanted to kind of give you that heads up a little bit. So make sure that you're going in and looking at the sliding filament model, but we'll talk about it here a little bit. But it is hard for students to understand sometimes. So skeletal muscle fibers are long, and they're cylindrical cells, and they have all these nuclei that we talked about, right? And then we basically the muscle fiber plasma membrane, we call that sarcolemma. And of course that muscle fiber cytoplasm we call the sarcoplasm. Also they contain what we call these glycosomes for glycogen storage as well as myoglobin for oxygen storage. And there's some organelles all through here. So these are all the microanatomy parts that's going to come together to create a sliding filament model. So these modified organelles, we have myofibrils, we have the sarcoplasmic reticulum, we have T-tubules, and so on. And of course, looking back, we've seen this before, but this is just sort of that microscopic anatomy of that skeletal muscle. When I start talking about the sliding filament theory, you're going to hear a lot about A bands and Z bands and light bands and things like that. So when we talk about the striated muscle, you're going to see that it's divided into these bands. So basically, this is showing different fibril muscles. One myofibril extends out here. You see it extending out. But basically, we have these banding patterns. These you have dark bands. Clearly, you could think they're darker colored, so you have that dark band there. You also have this light band that kind of spans through that. And then you have nuclei. So you have all kinds of nuclei running throughout this, right? So these striations or these stripes that are formed from a repeating series of these dark and light. We can go back to that slide and you see it's repeating. You've got dark, light, dark, light, dark, light, over and over and over. So these striations or stripes form from that repeating series of dark and light bands. And this is really important in anatomy and physiology. This is kind of getting more into the physiology side, but of course we've mentioned the structure, what it looks like, and the anatomy. But A bands are dark regions. And then within that, there is what we call an H zone, or a lighter region in the middle of a dark A band. And then there's something called an M line of protein, which is myomasin, that bisects the H zone vertically. And then there's I bands. Those are those lighter regions, that Z disc, that line. It's a coin-shaped sheet of proteins on the midline of the light I band. So there will be some pictures of that too. This, of course, is just this microscopic anatomy sort of image of this. So this is a micrograph of two portions, basically muscle fibers. Note this, as it says, the light and dark bands. But these, of course, are the nuclei spread throughout. This is one fiber here. So this little branch here is one fiber. But what you see within here is that you see these little, and I'm sorry, my arrow is not small enough or pinpointy enough to point to it very directly, but you can see it with the line. You see this dark A band, and then you see this little light I band, right? So keep that in your mind as we talk about this. Now we're going to kind of take a different view of this, a little slightly different, but it's the same idea. This is just a small part of one sort of myofilament, one filament, that's responsible for the banding pattern. So what you're seeing in this last slide, of course, is a lot of them put together so that you see sort of that banding pattern overall. This is one myofibril enlarged to show how that banding happens. So you see here, this is what we call that sarcomere. I'm going to go back and remember. It's just hard for students to remember, but basically we have all of these different terminologies. But you're going to see, sorry about that. We call this, of course, that sarcomere. One sarcomere. You see there's some thick filament, and of course there are things called the I band, which we mentioned, remember, is that light band. A band is dark band, and it alternates, right? So this is that thick area is called the myosin filament. And then you see these dark bands and light bands. So a light band, or this I band here, has something called a Z disc. And you can see that sort of lining here in that kind of bluish color. A band, remember, is that dark band. It has something called an H zone, and you can see that H zone as it's kind of spread there. And then, of course, you have that alternating I band. What you'll see is this just alternates back and forth. Inside of this A band is what we call an M line. Now, why am I telling you all this, or why is your textbook asking you, or why is this important for students to know, whether you're going into nursing or biology or EMT or whatever it is you're going into? It's important because this process, all of these things, have a specific function to help contract a muscle. So if you take your arm right now and spread it out, and then ball your hand into a fist and then do like a bicep curl. When you're doing a bicep curl, right, your muscle here, all of these are sliding back and forth. And it's kind of this overall physiological process that's pretty amazing. So it's important to know because these will lengthen and shorten. So these Z disc, H zones, M lines, A bands, I bands, all of that are doing a specific response in order to sort of move, to basically be able to have that contraction that happens. So this is, again, an even enlarged, a greater enlargement. So here you see M line and Z disc that we've talked about and all of that. Again, it's this here zoomed in even more so that you can see you have these thin filaments, you have thick filaments, and then you have those, still have the Z discs, you have these M lines. I bands, they have these little thin filaments only. That H zone we mentioned before, which kind of goes along towards, in the area of that midline on that last thing. Nice slide. or thick filaments only. And the M line, again, of course, that's close to where that H zone is, right? It's a range, but the M line, of course, runs through that. And that is thick filaments that are linked to those accessory proteins. Then there's the outer edge of the A band, and those have things like myosin filaments and actin filaments running through those. So there's a ton of things going on in a skeletal muscle, right? So again, let's zoom in even some more. So this is just one thick filament. So this is a portion of a thick filament. There are these little myosin heads. I'm going to go back a slide. You could see a little bit of these myosin heads on these thick filaments too. So it's that same thing. We're just zoomed in even more. And we have this on each one of these are little binding sites, ATP binding sites. So you remember before we said ATP is needed to take that chemical energy and convert it into that mechanical form of energy, right? It's changed. So we have to have a lot of these, right? So the ATP can bind, giving us that chemical energy so we can turn it into that mechanical energy that we have. So just think of as you're doing a bicep curl, even if you're doing it right, I'm actually, when I'm teaching, I do it too. So I'm sitting here bending my arm as I'm talking to you. But as I'm curling my biceps, basically I've got to have a bunch of ATP rushing to these lovely little myosin heads so that my muscle fibers can contract. So when we think about certain actions that we're doing, there's a lot of microscopic stuff going on, a lot of chemistry and biochemistry that's allowing you to change that chemical force into that mechanical force. So again, this is the composition of the thin filaments. So some thin filaments, they have these two strands of actin. You can see sort of the actin running through. You also have, it's twisted into a helix. And there's some proteins, troponin and tropomyosin. And we're going to talk a little bit more about that. Your textbook will even go into more detail than I talk about here. But these also have some subunits that are this myosin binding site. So you have to have certain things that are going to attach so that the chemical reaction can proceed. So what is this relationship between that sarcoplasmic reticulum, these T tubules, these microfibrils of that skeletal muscle? Well, you see here, you've got all of these, you got mitochondria. One thing I haven't mentioned too much of is that muscle cells have a ton of mitochondria. Well, the reason they have a ton of mitochondria is that is where, we've talked about before, it's like the powerhouse of the cell, as most people refer. You're taking ATP and that chemical energy and basically converting it into a usable form, and then, of course, heat is released off. You've got to have a lot of those in muscle cells because muscle cells need to make that conversion quite a bit. Other cells have mitochondria as well, but our muscle cells have a ton of them, and that should make sense because it needs to basically, since the mitochondria has ATP, it basically utilizes ATP to create a useful, or to generate a useful form of energy for us. Muscles need to do that a lot, and so whereas I might have a cell in my body that doesn't need a whole lot of energy all the time, but muscles do. So they're going to have lots of mitochondria. You've got all of these little muscle fibers coming through, and then, of course, there's these T tubules and these terminal cisterns, and you can kind of see it's protected as a layering over that. So not only do you have all of these I bands with Z discs, A bands with an M line and H zone, and alternating light and dark bands, you have a interconnectivity of myofibrils and T tubules of that sarcoplasmic reticulum so that you can perform these functions. So it's very complex and complicated with regards to how many steps there are. So when I mentioned several slides back about the sliding filament model, all of those components we've been talking about basically come together to allow contraction to happen. So you have this movement where it slides back and forth. Think of that bicep curl. You can put your hand on that bicep and basically feel that contraction. So the activation of cross bridges that generates that force. So shortening occurs when tension is generated by those cross bridges on those these thin filaments, and when it exceeds those forces that are opposing the shortening. And then contraction ends when those cross bridges become inactive. You no longer have that activity, and therefore that's the completion of that sliding filament model. So in the relaxed state, thin and thick filaments overlap only slightly at ends of the A band. The sliding filament model of contraction will state that during contraction, these thin filaments slide past those thick filaments, and that causes actin and myosin to overlap. So neither thick or thin filaments change length. They just overlap more, which is really hard for students to understand. It's sliding them. It's not stretching them. It's just sliding them over each other. And so when the nervous system stimulates that muscle fiber, these myosin heads are basically allowed to then, because they slide over one another, allowed to bind to actin, and they form those cross bridges. That causes that sliding process to begin. And so then that cross-bridge attachment forms break several times, each pulling thin filaments a little closer until it towards that center of that sarcomere in what we call a ratcheting action. So if you think of a ratchet and how, you could turn it, but you could have to hold it in that place, and you hear a little and then you pull it again, and go back. It's that same kind of thing. It's almost like if you're laying on if you have kids, or if you think about when you were a kid, or even if you've done this even recently for some reason, you're laying on the floor and you take your hands and you pull by grabbing onto something, and it pulls your body forward. But then you have to reach out again with your arms to grab something and pull your body the rest of the way, like a slithering motion. This is the same kind of idea. So Z discs are basically pulled towards the M line. That causes the I bands to shorten. These Z discs becomes closer. The H zones disappear, and the A bands move closer to each other, and that action can be finished or completed. So this is a fully relaxed sarcomere fiber. This is a fully contracted sarcomere fiber. So do you see that difference? One is basically relaxed here. One is contracted. So first, let's look at the micrograph at the top. You see this darkened bands. You've got these light bands, dark bands. You see that H zone and that M line in between that. And now see that difference. So looking at this Z line, you see that relaxed is sort of way out here. So this is where it was. Now this is where it is. So you can kind of see what's happening with that line and why it's important for us to understand that. Now let's look at it from this cartoon drawing image to see what it's like. Again, there's your Z line. This is where the Z line was. It is now here. So it's sliding this over so that these filaments can be activated with actin and myosin. So you can kind of see that difference. You even see all of these change, as that contraction happens. So it's important to understand how this happens, because it's where that chemical process begins and happens so that we can get that mechanical form of the energy. So that sliding filament model of contraction is really important to understand. So when we talk about contraction, of course, the decision to move is sort of activated by the brain, and that signal is transmitted down the spinal cord to the motor neurons, where it activates those muscle fibers. So neurons and muscle cells are excitable cells capable of action potentials, and of course that produces potential voltages. So actin potentials that crosses from the neuron to the muscle cell activate the neurotransmitter acetylcholine, and this of course is an abbreviation for acetylcholine. And then, of course, there's some ion channels that play a role. So the major role of changing membrane potentials, this is also where physics comes in, because we're actually now talking about voltages and ion channels and all of that. So there's two classes of ion channels: the chemically gated ion channel, and basically they're opened by chemical messengers such as acetylcholine or neurotransmitters. And then there's voltage-gated ion channels, which open or close in response to the voltage changes in membrane potential. So the anatomy of these motor neurons in this neuromuscular junction, skeletal muscles are stimulated by somatic motor neurons. So axons, well, we'll talk about more about these when we get to the nervous system, but it's important to know now how it works with our muscles. But axons, these are these long thread-like extensions of motor neurons that travel from the central nervous system to the skeletal muscle. And each axon divides in many branches as it enters into the muscle. So that axon branches that end on the muscle fiber, forming what we call a neuromuscular junction. Essentially, the connection or junction between the muscle and the nerve. So that neuromuscular junction, or sometimes is referred to as a motor end plate, same thing. Each muscle fiber has one neuromuscular junction with one motor neuron that is sending that signal for that muscle to move. So you can kind of see this is that overview of that skeletal muscle contraction. You see this is the axon coming out of that neuron, or that motor neuron, and you see that it's one of them, but with that multi-branching sort of creating this neuromuscular junction. This is, again, like that region that of that motor neuron contracts that skeletal muscle. And there's lots of axon terminals that kind of run through that. When you see that junction, when there is a connection between, you see here there's some junctional folds of the sarcolemma, and then, of course, there's what we call the synaptic cleft that runs through. And, of course, what you have in this neuron is a bunch of acetylcholine and neurotransmitters that are there, and they are released when they basically are touched. So you see these neurotransmitters are released into that synaptic cleft, giving that signal so that muscle can contract. So nerve comes to that muscle, releasing those neurotransmitters. I can't play this in here. You won't be able to hear it, but definitely listen in the main PowerPoint to this here, which is what the direct events at that neuromuscular junction. Again, also listen to the excitation-contraction coupling that occurs, since I can't play it in this. And then that kind of brings us to the muscles and the muscle tissues part, sort of the next step here. Okay, so moving on into more of that muscle tissue and how that works and its functions, we're going to kind of continue on. We've been talking about muscle contractions and how that works. So now we're going to sort of talk about different types of contractions. So the same principles apply to contraction on whether it's a single fiber or the whole entire muscle. So that contraction produces this muscle tension, that force is exerted on load or the object to be moved. And so basically the contraction may or may not shorten that muscle. So you have an isometric contraction. That's where no shortening has happened. The muscle tension increases but does not exceed the load. And then the isotonic contraction. And that's where the muscle shortens because the muscle tension exceeds that load that's there. So the whole muscle contraction, so the force and duration of a contraction vary in response to stimuli of different frequencies and intensities. So each muscle is served by at least one motor nerve. A motor nerve sort of contains of axons and up to hundreds of motor neurons. And those axons branch into terminals. And each forms what we call, and we talked about just a second ago, that neuromuscular junction with that single muscle fiber. So that motor unit is that nerve muscle functional unit. So a motor unit consists, again, of motor neuron and all those muscle fibers, 4 to several hundred it supplies. Smaller the fiber number, the greater the fine control you have. And then, of course, muscle fibers form a motor unit and are spread throughout the whole muscle, so stimulation of that single motor unit causes only a weak contraction of that entire muscle. So this, of course, is that motor unit that we're talking about, that one motor neuron and all the muscle fibers it innervates. So, of course, here you see that spinal cord, a cross-section of the spinal cord, and then, of course, you have that motor unit. This is motor unit 1 running and innervating there, motor unit 2 branching here. So axon terminates at that neuromuscular junction. And then you can kind of see what that looks like under a photomicrograph. And so you can kind of see that connection and how that works. So the muscle twitch is that simplest contraction. That sort of results from the muscle fiber's response to a single action potential from a motor neuron. Muscle fiber contracts quickly, then relaxes. And of course, the twitch can be the twitch can be observed and recorded as a myogram. So we can line record that contraction activity. So there's three phases of a muscle twitch. There's a latent period, basically the events of excitation, contraction, coupling. There's no muscle tension seen. Then we can see a period of contraction. That's that cross bridge formation that we talked about earlier, and that tension increases. And then, of course, the period of relaxation. And that is where ionic calcium, so Ca2 plus is what we call ionic calcium, it's re-entry into that sarcoplasmic reticulum, and that tension declines to zero. So that muscle contracts faster than it relaxes. So you can see what a muscle twitch looks like. This is a myogram where we're seeing each different thing happens. You have one single stimulus, this latent period, a little bit of a delay, and then that period of contraction comes way up. And then when that calcium starts leaching, back, re-entry into that sarcoplasmic reticulum, slowly, that period of relaxation. Now, this is still milliseconds, but you can see that period of relaxation takes longer, even though this is in milliseconds, than it does to contract. So that's that twitch. Different muscles have different twitch sort of lengths, right? This is still milliseconds. This is comparison of twitches in three different muscles. So the lateral rectus, basically you can see there's that delay period and it goes whoosh, but it actually comes down rather quickly. And then the gastrocnemius, basically you see delay period, of course, comes up, but it's so slowly going down. Soleus, while it still takes, it's slower coming up in that sort of that contraction period, but still the relaxation period is still a little bit longer. So each muscle, different muscle, has a different twitch or a different length of that twitch. So basically, how is that energy sort of distributed during different activities? So if you have a short duration, high intensity exercise, let's talk about running. You're talking about running quickly, sprinting. You have ATP stored in the muscles are used first. That's, good 6 seconds. 10 seconds, ATP is formed from that creatine phosphate and ADP through the process of phosphorylation. Then the glycogen stored in muscles is broken down into glucose, which is then used to generate ATP. So if you have already formed ATP for about 6 seconds, use that. It's used first. Of course, ATP in the next 10 or so seconds, we have if we have the components of ATP present, we make it really fast and utilize it. But after some time, we have to actually break it down and break it down to glucose and generate using that anaerobic pathway. And so by the end of exercise, we're working for that, right? And this, of course, is high intensity. Prolonged duration, let's say you're jogging for hours. ATP is generated by the breakdown of basically several nutrient energy fuels in that pathway. It's not necessarily going to just utilize ATP from that glucose. It's going to find it wherever it can. So you have these marathon runners, ultra-marathon runners, anything that lasts for hours of time. It's going to be taking fuel from a wide variety of places, wherever it can, so that it can generate that ATP that it needs. So some people say, well, high intensity, you might hear, especially if you're in the health sciences area, you might hear, this short duration increases cardiovascular fitness, right? It does, because you're telling your body, you better store some more full ATP, because if this chick does this again, basically we're gonna kill over. So we need to build up our muscles. So if she does it again, we can do it. Here, the body, of course, it's going to improve cardiovascular fitness, yes, but it's also going to lose fat. It's going to lose other things because it's breaking it down from different areas. So if you ever hear somebody say in weight loss, you need cardiovascular fitness, that short duration, high intensity exercise, and you need some prolonged duration exercise. It's true. Because here, you're not utilizing that fat cell. You're taking every bit of glycogen you have, or ATP you have, and utilizing it really quick. You still lose some fat here because it's at the end of the exercise, your body has to replenish it, and it gets it from a lot of places. But you should do a combination of both so that you have that full spectrum sort of use of that energy resources. Muscle fatigue, that's that physiological inability to contract despite continued stimulation. That includes sort of that low ionic balances, meaning, potassium and sodium and ionic calcium. There's not enough, there's low levels of that or high levels of that, too much, too little, and it disrupts that membrane potential of that cell as it crosses that membrane. You can also have increased inorganic phosphate. That means basically you can't really break it, that breakdown kind of, I hate saying it twice, but there's a breakdown. I was going to say the breakdown of that breakdown system. But it interferes with that calcium release from that sarcoplasmic reticulum, which hampers the overall power, and that fatigues that muscle. So there is some excess post-exercise oxygen consumption. So basically, for that muscle to return to its pre-existing state, oxygen reserves are replenished, lactic acid is reconverted to pyruvic acid, glycogen stores are replaced, ATP and creatine phosphate reserves are resynthesized. And so all of this is you're still working, exercising when you're done exercising, so to speak. So we have to replenish those steps that require extra oxygen, usually referred to EPOC, which is excess post-exercise oxygen consumption. It used to be called as oxygen debt, but those are some of the ways you might hear that. So the next sort of area, what are some factors that increase the force of skeletal muscle? Of course, high frequency stimulation, large number of muscle fibers that are recruited, large muscle fibers, all of these things that contractile force will cross bridges attach, and so that increases that force. So there's some velocity and duration of contraction. How fast the muscle contracts is influenced by the type of muscle it is, the load that's present, and the recruitment that's there. So muscle fiber type, classified according to two characteristics. You can have slow or fast fibers. That's the speed at which myosin ATPases split ATP, the pattern of electrical activity in motor neurons, and then, of course, you can have these metabolic pathways. Which metabolic pathway is used for that particular contraction? Is it oxidative fibers that's using aerobic pathways, or is it glycolytic fibers that are using anaerobic? And so that makes a difference. So based on these two criteria, skeletal muscles can be classified into three types. They can be slow oxidative fibers, fast oxidative fibers, or fast glycolytic fibers. So most muscles contain a mixture of fiber types, so that sort of results in a range of that contractile speed and resistance. But all fibers in one motor unit are the same type, and of course genetics will dictate the individual's percentage of each. So your genetics will play a role in what type of, at least the numbers or the distribution of the different types. Everybody has all three, but there's a different makeup of those. And you can imagine, that can lead to it being harder for people to control weight. It could be their motor skills and different things like that can vary from person to person because the makeup of someone is these different fibers are based on genetics. So those factors, again, of course, the predominance of these fast glycolytic fibers, whether something is small load or increased movement, the predominance of those slow oxidative fatigue-resistant fibers will actually affect whether duration and velocity of those. So basically, you can see the influence of load duration and velocity during muscle shortening. So you can kind of see here, you have the greater the load, the briefer the duration of the muscle shortening. So the more the load, so distance that is shortened, and then of course the time on light load, you can see that arch is much higher. When you're on a heavy load, you can see basically that distance is short. It has less shortening compared to that light load. And then, of course, that increased load, the greater the load, the slower the muscle shortening. So adaptation to exercise. I mentioned this a little bit a couple slides ago, of course, but I kind of want to mention it again here as well. Aerobic exercise or endurance, basically jogging, swimming, bikes, leading to increased muscle capillaries, number of mitochondria, and myoglobulin synthesis. So it is very true that, I've had students say, wait, we get more like little capillaries of blood flow to a muscle if I exercise? Yes. People, some students have said to me, I thought everything was sort of static, like it was just there. No. The more you go through aerobic exercise, and the more that you're doing, the better you have with the flow of blood, the distribution of oxygen, because you actually grow more capillaries to those muscles, meaning more oxygen, more blood flow. You increase that number of mitochondria. So the more you work your body, the more your body says, uh-oh, I don't know what this person is doing to me, but I better be more efficient at breathing. And that's why over time, it gets easier. I'm a runner. I love to run. However, I'm the type of runner that I guess maybe with me getting older, I might have more minor injuries. No major injuries, but I might, I might have some runner's knee or something happen more often. So I might take a break from running because I don't want to have permanent damage, right? And so I'll run and I'll be doing great, and all of a sudden I will have an issue with my knee. And I'm like, oh, well, I better take a few days off, maybe even a week or two off. Well, at one time I could, before I had the injury or the sore knee, I could run an hour straight, for example. I just run. I take a week or two off, and I come back to it, and I can't really run that hour anymore. And that's because my muscle capillaries have started to decrease. The number of mitochondria have decreased and all of that because my body didn't need it. I wasn't utilizing it, so now my body said, man, she doesn't need this anymore. Why keep spending the effort to make all this stuff? But then when I get back into running and run for a few weeks, my body says, oh, here's the crazy lady doing it again. Let's build it up. So your body will adapt to exercise in both directions. When you don't exercise, your overall fitness decreases because you're not as effective at getting oxygen and blood because you don't have as many capillaries anymore. You don't have as many mitochondria. You don't have those things. And so, but then when you do exercise, you build those up and it becomes easier over time. Resistance exercise, of course, typically anaerobic, that's weightlifting or isometric exercises. That leads to muscle hypertrophy, usually primarily to increase an increase in fiber size. You get increased mitochondria, increased muscle size, and strength. And so that resistance exercise is very important too. Not only does it help you later when you're off to do if it's cardiovascular fitness, the better resistance training you've had, the easier it is for your body to adapt to that. So it's important to have both. Now, kind of moving into smooth muscle. Smooth muscle, of course, is found in the walls of most hollow organs. And you see these in things like respiratory, digestive, urinary, reproductive, circulatory, except in the smallest of blood vessels and except the heart. Again, it's not found in heart because the heart contains cardiac muscle and it's not smooth. Most smooth muscle is organized into sheets or tightly packed fibers. And so most organs contain two layers of sheets. There's a longitudinal layer where the fibers run parallel to the long axis, and the circular layer which runs around the circumference of the organ. So the longitudinal layer, that contraction causes the organ to shorten, and the circular contraction causes the lumen of the organ to constrict. These alternating contractions and relaxations of these layers mix and squeeze substances through the lumen of hollow organs. Your stomach is a primary example. If you ever I know when I teach anatomy too, I have a video that shows this, but when your stomach is digesting food, it just churns and it just curls over and rolls, and it's really kind of amazing. Most people find it gross, but it just kind of contracts and rolls and squeezes and all of this, and it's constantly moving. I hear people say, "Well, is that my belly growling?" A little bit. That sound that's the movement of those liquids as it's being squeezed. But your stomach even is still moving when it's not. You don't hear that growling sound, and so you have this constant kind of bending and folding, and so that's what smooth muscle is doing for us. Of course, smooth muscles are these sort of spindle-shaped fibers. They're thin and short compared to skeletal muscles, which are wider and longer. Only one nucleus with no striations. It lacks that connective tissue sheath. It contains endomysium only. Remember, there were 3 in skeletal muscle. Here we only have that one. There is still innervation, of course. So you can kind of see this is the autonomic nerve fibers that innervates those smooth muscles. And of course, there are those synaptic vesicles. There's mitochondria, things like that are present too. It's just a little different than skeletal muscle. Smooth muscle has less sarcoplasmic reticulum and no T-tubules. So sarcoplasmic reticulum is then less developed in skeletal muscle. And so it's less developed than. But sarcoplasmic reticulum does store that intracellular ionic calcium, but most calcium used for contraction has extracellular origins. And the sarcolemma contains this pouch-like infolding called cavitae. And cavitae contains numerous amounts of these ionic calcium channels that open to allow this rapid influx of extracellular ionic calcium. Intermediate filaments and dense bodies of smooth muscle fibers harness and pull. That's generated by myosin bridges. So you still have that movement, but you don't have that sliding filament theory in the same way as you do with skeletal muscle. You do have thick and thin filaments. Those myofilaments are spirally arranged. So smooth muscle sort of contracts in a corkscrew-like manner. You heard me say when the stomach, it churns and rolls, and that's that kind of movement that you're going to see with that as it contracts. Intermediate filament, dense body network is located in this type of muscle. So you can kind of see this contains this lattice-like arrangement of these non-contractile intermediate filaments. They resist tension. You have dense bodies that are located here as well, and the proteins that anchor to the filaments to the sarcolemma at regular intervals, and they correspond to Z-disc of skeletal muscle. So during contraction, these areas of sarcolemma between dense bodies bulge outward and make that muscle cell look puffy. And so you can see here is that contracted smooth muscle fiber. So you can kind of see kind of crunched in there, and then there's that relax. So relax, sorry, I have instead of going back and forth really quick, I have a line in between it. Relaxed, and then, of course, contracted. And maybe I could do it better if I just click on that. There we go. So relaxed, contracted. So we've seen this before already. We saw this at the very beginning of the chapter, but you can kind of see this comparison of skeletal, cardiac, and smooth muscle. Whoops, I'm sorry, guys. So basically, you see that difference. We talked about it before. You do see the comparison again here and what it looks like. Again, looking at skeletal muscle, you see this bands here. In cardiac, it's a little different. You do still have Z-disc, not as complex. And then, of course, smooth muscle, we don't have this cavitae at all. Or only cavitae. And then, of course, you can see the different innervations that are happening, so make sure to review this over and over. So skeletal muscle, of course, you see that direct there. Cardiac, going straight to cardiac tissue. And so it's a little bit different in each, but all serving similar purposes as well. Also, the comparison of speeds of contraction and, of course, the site of calcium regulation. So you can see how different those are in each of those. But you can also see the different speeds of that contraction as well. And so that contraction of smooth muscle, energy efficiency of smooth muscle contraction, it's slower to contract and relax, but it maintains the contraction for longer periods of time using less energy. And so that's important because we want our muscles, things like our stomach muscles, to be able to churn and break things down. So there's slower ATPases. The microfilaments may latch on together to save energy. So most smooth muscles maintain a moderate degree of contraction constantly without fatiguing. That's referred to as what we call smooth muscle tone. So it makes ATP the aerobic respiration pathways. There is still regulation of contraction like we had in skeletal muscle. You have that you're still controlled by nerves and hormones and chemical changes. That neuroregulation results in increases of ionic calcium in the sarcoplasm. The responses depend on that neurotransmitter that's released and the type of receptor molecules as well. Hormones and local chemicals. Some smooth muscle cells have no nerve supply. They depolarize spontaneously. And chemical factors can include hormones, high carbon dioxide, pH, low oxygen. And so some smooth muscles respond to both neural and chemical stimuli. There are some special features, of course, of smooth muscle contraction. It has a response to stress, stretch, stress relaxation response. Basically stretches only briefly and then comes back and adapts to that new length. And then length and tension changes, basically can contract when between half the size and twice at its resting length. So that allows the organ to have a huge volume changes without becoming flabby when relaxed. So it's allowing that tension change without stretching it too much. When we're talking about myoblasts and things like that, basically when we're talking about, and I'm not sure why this ended up down here. Let me take this out because it got skipped down from skeletal muscle. I just saw that. Sorry about that. So if you have questions, please let me know. Make sure to go in and watch those videos, especially the videos in the PowerPoint and the extra stuff of the sliding filament theory. That's going to be very, very important to understand. But if you have questions, of course, after you review resources and all that, if you have questions, please feel free to reach out. I'm always happy to help and hope you have a great day.